Hey everyone, Professor Spencer here. In this video, we're going to talk about action potentials. This is a really important concept to understand and one of the more difficult concepts to wrap your head around. The goal of this video is to present this topic in a simple form so you can have a strong foundation for your exams. You'll build upon this knowledge in other places in the body where similar things are happening, like in cardiac muscle contraction. Alright, let's get started. There are two main modes when we're talking about action potentials in a neuron. We have the excitatory mode and the inhibitory mode. Each mode is triggered by the arrival of a different neurotransmitter. The excitatory mode is triggered by the arrival of an excitatory neurotransmitter like glutamate. The excitatory mode is engaged in order to get the neuron to fire the action potential. The inhibitory mode is triggered by inhibitory neurotransmitters like GABA. This inhibitory mode is engaged when we want the neuron to not do anything or not fire an action potential. First, let's talk about the excitatory mode. And this mode is what causes the neuron to fire the action potential. This is the most important part, and then we'll talk about the inhibitory mode. Let's start by setting the stage. Here we've got our neuron. It looks kind of funny because I've exaggerated some of the proportions in order to have enough room for the animations. In green, we've got the sodium channels, and in red, we've got potassium channels. We're using these colors because generally green means go and red means stop, and you'll see why in a second. When a neuron is just hanging out waiting for a stimulus, the charge inside of the neuron is slightly negative relative to the outside or the interstitial fluid. The outside of the neuron is slightly positive. We've also got a lot of sodium ions hanging out outside the neuron and a lot of potassium ions inside the neuron. Each of these ions want to go down their concentration gradient, so sodium wants to enter the cell and potassium wants to leave, except they can't. Each ion needs to pass through its own specialized channel. Many of these channels are closed by default. Now there is another type of potassium channel called a leak channel. These leak channels allow potassium to diffuse to the outside of the neuron. We also have the sodium-potassium pump, which uses ATP to pump out sodium and pump in potassium. Keep this in mind because it will be important later. What I want you to take away from all of this is that the sum of all these parts maintains an average voltage of the neuron at around minus 70 millivolts. This is what's called the resting membrane potential. A change in the concentration of either ion will cause the voltage of the neuron to change. And when we have a change in voltage, that's when things get interesting. Let's begin with our excitatory mode. In order to get something to happen down here in our axon terminals, we need to initiate something up here in our dendrites and soma. Let's activate our excitatory mode with the arrival of the excitatory neurotransmitter, glutamate. Glutamate kind of acts as a key to open the hatch on these sodium channels. So when these channels open up, sodium can move down its concentration gradient into the neuron. The fact that we need a substance to open these channels makes these ligand-gated channels. When these channels are open, sodium enters the cell and causes the neuron to become more positive. This slight change in charge is called a graded potential. Because we're making the neuron more positive, this graded potential is specifically called an EPSP, or excitatory postsynaptic potential. Notice on our graph, our voltage has changed slightly, but it's not enough to create an action potential yet. So what do we need? We need more sodium. Let's send a few more glutamates in. These glutamates open up more sodium channels. Now we've got more sodium coming in, and our charge is becoming more positive. By the way, the combined voltage change from all of our new sodium coming in is called summation. All of this new sodium entering our neuron has caused our voltage to increase to minus 55 millivolts. Minus 55 millivolts is what's referred to as the threshold. This is important because this triggers more sodium channels to open, which are located at the axon hillock. These sodium channels at the axon hillock are a little bit different than the ones we have in the dendrites and soma. These sodium channels don't need a neurotransmitter to open them. They only need a certain voltage, which is minus 55 millivolts. Therefore, these channels are called voltage-gated channels because they're triggered to open at a specific voltage. So our first channels at our axon hillock are opened and an action potential is born. Sodium will enter the neuron through these voltage-gated channels, raising the charge of the adjacent area to minus 55 millivolts. This causes the adjacent voltage-gated sodium channels to open next. And this continues like a domino effect down the axon. This is how an action potential moves down the axon. You can imagine that with all these voltage-gated sodium channels open, a lot of sodium will be entering the cell. You can see this on our graph, and it's rapidly making our neuron much more positive. This rapid rise in positivity is called depolarization. Depolarization. So much sodium has entered, in fact, that our charge will rise all the way up to positive 30 millivolts. And at this point, voltage-gated sodium channels are programmed to shut. They won't let any more sodium in, and depolarization has now ended. So how does the neuron get back to minus 70 millivolts? Well, remember we've got all this potassium inside the neuron, and potassium channels in the membrane. At positive 30 millivolts, these potassium channels are triggered to open, and this lets potassium flow out of the neuron down its concentration gradient. Remember, potassium is also positively charged, but moving that potassium out of the neuron causes the neuron to become negative again. You can see this change on our graph. This drop in voltage caused by potassium ions leaving the neuron is called repolarization. Repolarization. Interestingly, voltage-gated potassium channels are slower to close than voltage-gated sodium channels. This causes the neuron to, in a sense, get rid of more potassium than it needed to in order to restore its default voltage of minus 70 millivolts. Instead, the neuron dips all the way down to minus 90 millivolts, and this is called hyperpolarization. Great, our neuron is back to its negative charge, but wait a minute, something's not right. We now have more sodium inside the neuron and more potassium outside of it. This is the opposite of what we started with. How do we get it back to our original ion concentrations? Well, remember from the beginning of the video when we mentioned the sodium-potassium pump? This pump is what restores our concentration of ions. Using ATP, the sodium-potassium pumps send out the sodium from within the neuron and take in potassium from the outside. This gradually restores our concentration of ions so that the neuron can fire again. Okay, we've talked about how the neuron fires an action potential. Let's briefly talk about inhibitory mode. This is going to be very quick because there's a lot less to it. So let's reset our neuron back to its default state or resting membrane potential. Now let's say our neuron receives a different neurotransmitter. Instead of glutamate, we get GABA, which is an inhibitory neurotransmitter. GABA binds to ligand-gated potassium channels. As we saw before, potassium wants to diffuse out of the cell. So when potassium channels are open, potassium's leaving the neuron, making it more negative. These drops in voltage are another form of graded potential known as the IPSP, or inhibitory postsynaptic potential. And you can see this on our graph. This drop in charge makes it harder for the neuron to become positive and fire an action potential. It gets it further away from that minus 55 threshold potential. And that's the gist of it. Remember, sodium entering makes the neuron go. Potassium leaving makes the neuron stop. Remember, repetition is key to success at A&P, so rewatching this video will definitely help it make more sense. Also, here's a handy graph for your reference. Be sure you can draw this graph and understand what is going on with the neuron at each phase. This will really help you on exams. If you enjoyed this video and found it helpful, don't forget to action potential the like button and subscribe for more A&P videos just like this. I'll see you next class.